There we go. Perfect. This is the outline of my presentation. So I'd like to run through the utility of autoantibodies and discuss briefly our technology and give specific case studies for anti-drug antibody response with Humira, diagnostics in cancer, some consideration of prediction of drug responses, patient stratification in autoimmune diseases, and some recent developments in Parkinson's disease. So here you have a brief overview of the company. We have a broad portfolio of customers, 9 of the top 10 pharma companies. We work with research foundations such as the ONJCRI and Michael J. Fox Foundation, and leading universities. Our founder, Jonathan Blackburn, sits on various HUPO and FNIH biomarker committees. And you can see we're a fairly well-established company. We were a Cambridge University spin-out back in 2008. And in 2016, we opened our protein production and high throughput assay facility. A couple of years ago, we obtained a major equity shareholder, and that's allowed us to expand. And we're in the process of commissioning new facilities in Boston. So biomarkers are potentially really valuable tools to identify disease, to classify patients as responders or non-responders, and perhaps to stratify patients for clinical trials. However, all biomarkers are not the same. DNA, RNA, and proteins are macromolecules that provide a snapshot in time and have real value. However, the mood at the Biomarkers UK conference last year, and actually, thank you, Jackie, for also highlighting this, is that these approaches have largely failed to deliver on their promise. And we at Sengenics believe that the human immune system might be able to do a remarkable job of marking out aberrant disease-associated changes in proteins through the production of autoantibodies, and that these autoantibodies might provide a better class of biomarker that is more specific and more distinct to the disease state than DNA and protein markers have provided thus far. So autoantibodies are thought to arise in disease for a number of different reasons. It could be over or inappropriately expressed. They could be produced from variant genes, aberrantly post-translationally modified, aberrantly proteolized, or contain mutated epitopes. And disease tissue misregulation can lead to unique patterns of neoepitopes. And it's likely that different routes will be significant in different disease states. So with that said, autoantibodies make really great biomarkers because they're disease specific. They manifest early in disease, they are stable, abundant, and easily accessible. I'd like to take a moment to note that autoantibodies like to bind to folded protein structures, with the literature saying that roughly 90% of all B-cell epitopes are discontinuous, surface exposed, and typically hydrophilic in nature. Anybody who has ever run a Western blot knows that antibodies can also bind non-specifically to exposed hydrophobic surfaces on unfolded proteins with very untidy results. So to identify physiologically relevant autoantibodies, you really need to work with correctly folded antigens, which, as I'm sure many of you know, isn't straightforward across even modest collections of proteins. And this is where our technology comes in. So we have a proprietary process that allows us to print recombinant proteins on an array format for highly sensitive and specific detection of autoantibodies from patient serum and other sources. The proteins are chimeric proteins fused to the BCCP protein, bacterial biotin carboxyl carrier protein. And that acts both as a folding sensor and chaperone. And we use a proprietary hydrogel array surface. And these approaches eliminate the common problems of misfolded proteins and non-specific binding. We can print the antigens in multiple formats. And here you see our largest antigen array in a quadruplicate format. So the array proteins are stable for long-term storage, and the assays are highly specific with very high signal-to-noise ratios. The intra and inter-array reproducibility is exceptional, and the sensitivity of detection is down below 10 picograms per mil. So here are our standard products. Most projects start with the discovery platform that we call iOme. And indeed, all the case studies I will show you use this array. In addition, we have a cancer testis antigen array, the CTA array, and the P53 array with 100 clinically relevant P53 mutations. And we have a SARS-CoV-2 screening array. And finally, we have the capability to design and manufacture bespoke custom arrays. And in fact, this is the journey of most of our projects. The discovery phases uses iOme, and then a more focused custom array is designed for further investigation and validation. And it should be noted that we offer two modes of operation. Most customers send samples to us, and we process the samples and return fully annotated data. And we have a finely tuned bioinformatics pipeline for this analysis. It is also possible to purchase the arrays to run in-house in your own labs. And so this summary slide shows what we have and where we're heading. We can offer autoantigen identification. We can offer post-translational modifications, such as citrullination. We can detect all isotypes and subclasses of autoantibodies and offer a dual color detection. And our future will include assays for Fc effector function. So now let me offer some examples of the discovery that is possible on Syngenix arrays. So let's take a look at drug response prediction. This study was published last year, 2021, and is a Humira study. It comes on the tail of a smaller 2016 study showing that rheumatoid arthritis patients treated with infliximab can raise an immune response against this biologic, and that anti-Ro SSA antibodies, autoantibodies, associate with this response. And it should be noted that anti-drug antibody positive patients are also poor responders to adalimumab based on their EULAR score. So the study comprised 4 healthy controls plus 30 anti-drug antibody positive and 32 anti-drug antibody negative patients. There were no significant differences in the positive rate of rheumatoid factor or anti-citrullinated peptide antibodies, dosage of corticosteroids or methotrexate between the anti-drug antibody positive and anti-drug antibody negative patients. Serum was collected and assayed on the Syngenix iOme array and gave rise to a panel of biomarkers associated with the presence of anti-drug antibodies. From the initial cohorts, we see here the top 20 putative markers correlating with anti-drug antibody or poor EULAR response in plasma samples at baseline and week 24 of Humira treatment. The expression level of the differentially expressed autoantibodies shows quite clearly responder and non-responder groups. And you can see at the bottom here, clustering together, in purple are the anti-drug antibody negatives, and in blue, co-segregating are the good EULAR responders. So the best subsets of markers were selected based on a backward selection iterative process known as recursive feature elimination. The best panels were then stability tested by random forest analysis, and individual ROC curve analysis of the putative biomarkers revealed that anti-TROV2 antibody at baseline and at week 24 had a high discriminative power for predicting the emergence of anti-drug antibody and poor EULAR response. However, when the biomarker panels were assessed, although TROV2 and SSB were found in all panels, the diagnostic performances, particularly of this panel of 8 biomarkers, was found to be superior. And finally, the authors note that we are the first to use a high-density protein microarray with machine learning approach to identify protein biomarkers that are predictive of anti-drug antibody development and therapeutic response to adalimumab. And it's worth reiterating here that this biomarker panel pre-existed in patients prior to their exposure to adalimumab. So let's take a look now at biomarkers for early detection of melanoma. This study was published in 2018 by Pauline Zenker. They looked at 124 stage 1, stage 2 melanoma patients and a similar number of controls. They identified 139 biomarkers, including some that were previously associated with melanoma. And they further defined a 10-marker panel with a strong AUC value and sensitivity specificity values of 84 and 79%. An unpublished continuation of this study of stage 1, stage 2 melanoma reveals distinct subgroups in the cancer cohort. And you can these subgroups plausibly represent distinct molecular subtypes of melanoma with different etiologies, disease trajectories, and different treatment responses. We know that this work is under continuing study. So in a similar large-scale study, a group from National University of Singapore identified 19 autoantibodies from early and late-stage NSCLC patients. And they refined this down to a 7-marker panel that had the power to identify and discriminate early and late stage NSCLC. And this study forms the basis of a patent from NUS. And it's worth noting that similar diagnostic biomarker panels are being developed for other cancers. This study was recently published. It's from the University of Birmingham, and it identified a prognostic survival signature for NSCLC patients. The cohort was 157 NSCLC patients who received the standard of care, this being surgical resection of the tumor. The cohort had 92 survivors and 65 non-survivors. They identified a 13-marker panel with a high predictive power for survival, as shown by the ROC curve analysis. And the Kaplan-Meier curves show an unambiguous outcome. Expression of this biomarker set has a poor prognosis. The hazard ratio for this panel is 19.6, and that compares very favorably with the IASLC staging prognosis method, which gives a hazard ratio of only 1.24. And again, this work is moving on to a follow-up validation cohort. So my final slide on relating to cancer concerns drug response stratification. So there are a number of studies that this refers to. One is from an academic group at Lausanne who are preparing the study for publication, and the others are all from internal projects. But unfortunately, I can't share significant details from either of those data sets. However, I can summarize the findings as follows. So the studies used several different checkpoint inhibitors. Patient samples were taken prior to treatment, so any predictive outcome is pre-treatment. In several studies, we were able to identify predictive signatures for whether patients will respond to the treatment or not, as shown in this branch point. So many anti-cancer biologics have significant immune-related adverse events, and we were able to identify predictive biomarker panels for these adverse events. And it should also be noted that IRAEs can manifest differently in different patients, and we were able to identify biomarkers that predict which organ systems would be affected. As you can imagine, this is a finding of great interest to our clients. So let me turn now to some autoimmune projects. In a recent Sjogren's syndrome project, we identified an autoantibody panel that identified primary Sjogren's from controls, and a similarly strong panel that discriminates primary Sjogren's from non-Sjogren's sicker. So you can see here we have a 10-marker panel that discriminates primary Sjogren's from the controls, with a sensitivity of 96%, specificity of 87%, and a 6-marker panel that discriminates primary Sjogren's from non-Sjogren's sicker, with sensitivity specificity values of 87 and 81%. And we can also stratify these two diseases from SLE. We included an SLE data set that we already had for that. Interestingly, within the primary Sjogren's cohort, we identified 3 subgroups which seem to correlate with clinical features. This was unexpected, and at this point, further investigation is needed to assess potential different disease trajectories and treatment responses. So this case study relates to a large-scale study of a lupus cohort that was published in 2018 by Miles Lewis et al. At Queen Mary's University, London. They used sera from a discovery and validation cohort of 277 SLE individuals and 288 age and ethnicity matched controls. And they assayed using the IOM array to identify novel autoantibody biomarkers. So they found 68 novel and 11 known autoantigens, and they refined that down to a panel of 26 autoantibodies. And you can see here, unsupervised clustering of the autoantibody repertoire reveals 4 distinct SLE clusters. So on the left, the clusters are identified by the pink boxes, and on the right, those clusters are reiterated. This is the validation cohort. And these clusters plausibly represent distinct molecular subtypes of SLE with different disease trajectories and different responses to treatments. And underneath, you can see we've correlated these subgroups with other clinical markers. So SLE1A, for instance, is strongly anti-TROV2, anti-SSB positive. It has high levels of ANA antibodies. SLE2 has a broad range of autoantibody positivity with high levels of anti-dsDNA. And S1B and 3 have lower levels of ANA and anti-dsDNA. The further analysis of these subgroups can be visualized like this, and the authors raised the interesting possibility that group 3 patients, shown here in blue, may be susceptible to B-cell directed therapies such as rituximab, whereas group 2 patients, shown in green, may be better treated with non-B-cell directed therapies. And again, the cohorts that was used in the study did not have the clinical data that could be used to further investigate those hypotheses, but we are trying to move that forwards. So the next few slides show some data from a project that we're currently working on. And this relates to a paper from 2017 on the Parkinson's at-risk study cohort. In this study, they reported the rate of conversion to a clinical diagnosis of Parkinson's disease in those at risk based on olfactory testing and dopamine transporter imaging, DAT imaging results. They identified that those with a DAT deficit, as judged by reduced beta-CIT SPECT imaging, have a high risk of phenoconversion to Parkinson's disease. They further noted that reduction in olfactory function, as judged by a standardized scratch and sniff test, was observed in up to 90% of patients who subsequently showed a DAT deficit and phenoconverted to Parkinson's disease. So the work I will outline is funded by a grant from the Michael J. Fox Foundation. The initial data set is derived from a cross-sectional study of the PARS cohort and has been designed in collaboration with the MJFF and with input from Ken Marek. So given the prevalence of Parkinson's disease in Western societies and the high cost and inconvenience of PET SPECT imaging, plus the observation that any therapeutic intervention to mitigate Parkinson's disease will have the greatest chance of success at the earliest possible diagnostic point, a serum biomarker that indicates the possibility of Parkinson's disease early in disease progression would be of interest. From our expertise in the field, we reasoned that there might be an autoantibody profile that satisfies this need. So here we have the study design. We were given access to 145 samples from the PARS cohort. No participants had dementia at the time of enrollment. The participants were segregated into hyposmic and normosmic, and the normosmic are essentially the control group here. And it's important to note that over the 4 years of the PARS study, none of this normosmic group phenoconverted to Parkinson's disease. Of the hyposmic group, they further divided into those with measurable DAT deficits and those with no DAT deficit. And of the hyposmic no DAT deficit group, none of them phenoconverted to Parkinson's disease during the 4 years of the study. Of those with DAT deficits, they divided into patients who phenoconverted to Parkinson's disease or those who were not diagnosed with Parkinson's. So the red eye here indicates the branch points at which we compared plasma autoantibody levels. We assessed whether it might be possible to identify hyposmia, whether that deficit could be assessed without PET SPECT imaging, and whether a biomarker profile exists for phenoconversion to Parkinson's disease. The data required are normalized and assessed by ROC curve analysis and by machine learning for significant differences between the groups. So here you can see the ROC curve analysis for 3 of the comparison points. The area under the curve values are good, as are both the sensitivity and specificity values. We assessed the appropriate biomarker panels, and they vary, and they're in the range between 10 and 31 autoantigens per panel per decision point. So you can see in the top left there, we have the hyposmia versus normosmia group. So we have a very good sensitivity specificity value to discriminate between those two. In the top right, of the hyposmic group, we compared the DAT deficit versus no DAT deficit branch points, and we identified a different biomarker set, so 31 different markers, again, with good discrimination between those with DAT deficits and those with no DAT deficit. And finally, here at the bottom, those hyposmic DAT deficit patients were then assessed who either phenoconverted or did not phenoconvert to Parkinson's disease. Their biomarker profiles were assessed, And again, we have some pretty encouraging data there. So here are the data overlaid on the study design map. And now these are early data. We are just in the process of completing the analysis, but we're not quite done with it. And this is a cross-sectional study as well. And we intend to validate the findings on the longitudinal cohort from PARS. However, I think it's clear that there exist autoantibody profiles that clearly allow screening for hyposmia versus normosmia, for the identification of patients with a DAT deficit. And in this case, these patients will be sent for PET SPECT imaging, at least initially. Stratification of DAT deficit individuals who are at high risk of phenoconversion to Parkinson's disease. In additionally, we've done some other comparisons between the hyposmia no DAT deficit group and the normosmia no DAT deficit group, and hyposmia no DAT deficit versus normosmia no DAT deficit. So in total, we've identified 161 proteins or autoantigens that form these various biomarker panels. We looked at the biological pathways and processes that our candidate autoantigens associate with this. In this instance, we've used Web Gestalt, and there are a wide range of paths and processes, which I haven't shown everything, but for us, some of the more interesting pathways are highlighted here. So that's all I can tell you really for the moment. Hopefully this has given you some insight into what is possible with autoantibody profiling and Parkinson's disease. And you'll forgive me, I hope, for not sharing the details of the proteins, but we will be submitting the manuscript very soon, and we hope that it will be favorably received. So in summary, autoantibody biomarkers have high value in early disease detection in all cancers that we've studied to date, in patient stratification, prediction of treatment outcome, disease monitoring, treatment monitoring, I should say, prediction of immune-related adverse events. And we think that some of these outcomes could be of interest to the development and testing of new and existing therapeutics. And additionally, we have shown that biomarker quantitation is possible from dried blood spots. And this opens the door perhaps to a population-based screening method or project. And so with that, I bring this to a close. Thank you for your attention, and I'd be happy to take any questions.